Renaissance age of information in general, right? Because it's the time in history where we have the Enlightenment, right? Where people are re-examining what they know and how they know. It's actually the other one that's empty. Sir? It's that one.
Oh.
So if you study any other aspects of understanding, like Western civilization or chemistry or physics or anything else, you're going to find this period of time is really a boom in learning for everybody involved. So we have that. We're going to talk about a couple of key things. There are bazillions of cool things that happened during this 200-year period. We're going to come back and talk about different ones when we talk about epidemiology. We'll talk about some more when we talk about immunizations and things like that. But here I've chosen 5 that are kind of key right now, just a general understanding of bacteriology and microbiology. And then I added these 2 from what people are calling maybe like a second golden age of microbiology. And this happens around the 1970s with the really a big explosion in modern biotechnology. During the 1970s, restriction enzymes were discovered, and so learning how to splice pieces of DNA was discovered, and all this neat stuff happened. Most of it has not a lot to do particularly with microbiology, but 2 things that I wanted to mention, and that we'll see, we're throwing out there because we're going to see them again later on in the class. So we're going to look through the golden age of microbiology, and then we're going to fast forward 100 years and look at 2 key advancements in microbiology in the 1970s and 80s. So first of all, this handsome chap here, Francesco Redi, who in 1668 began a debate that we are going to talk in depth about. Is really where we're going to focus most of our attention in this history of microbiology, but he began what is called the biogenesis debate. And you're going to find in a few minutes that that was a debate that was thought to have been settled since the time of Aristotle, right? You know, Aristotle, that was a long, long time ago, like BC, a long time ago, right? So for a long, long time, what Aristotle thought was that, you know, that was it. And then he thought, I don't know, like this is a little bit kooky. So he started this debate on biogenesis. And we're going to talk a lot about him in a few minutes, right? So he starts off our perspective on the golden age of microbiology, right? The book might start somewhere else, but who's writing your tests? I am. So who are you going to pay attention to? This guy, right? Then we're going to fast forward a little bit, right, to 1796, so over 100 years later. We're going to zoom through all sorts of really cool stuff and talk about Edward Jenner. Edward Jenner is the father of immunizations. He is the first person to ever put together scientifically the idea that you could inoculate someone with something that is less pathogenic in order to protect them from something that's deadly. And in his case, he found that if you inoculated people intentionally with cowpox sores, like this crust from a cowpox sore, which admittedly is disgusting, and you poked that into another person, right? Not a lot of ethics going on in the 1700s. Notice he's poking a little boy with that disease-causing organism, right? And you're like, well, cowpox, that's not such a big deal. But then he exposed him to smallpox, and he didn't contract smallpox. Now, this actually was a really ancient process. The idea of protecting someone against smallpox actually exists probably for 2,000 years, but the problem was that people were intentionally giving each other smallpox to protect them against bad smallpox later on, right? What happens sometimes when you give someone smallpox intentionally, you kill them from smallpox. So this was the first time that someone had clinically demonstrated that you could inoculate someone, right, inject someone, expose someone to something that was non-pathogenic, non-lethal, and protect them against something that was lethal. So we're going to come back and talk about Edward Jenner later on in our epidemiology unit, but we're going to give him a little nod here as part of our golden age of microbiology, right? Then we're going to fast forward to almost another 100 years to Louis Pasteur, who is probably the most famous name on this list, right? Depending how strange you are and what you like to read. You might have heard these other people. Most people haven't, right? But most people, at least pasture sounds familiar to them because we have this process of pasteurization, right? And a lot of our foods are pasteurized, which means that they have had the number of spoilage organisms reduced to a safe level, right? So like your milk is typically pasteurized, which means that when it comes to you, as long as you keep it in the refrigerator, you will not get sick from drinking that milk. Of course, if you leave it on the countertop, you will get sick because pasteurization doesn't sterilize milk, it just makes it safe for consumption, right? But among that and all sorts of other things that he did, and his name will come up a couple of times in this class, was he connected a protozoan, which is a microscopic eukaryote, just like those things that you looked at on Tuesday, right? So kingdom Protista includes the protozoans, so the volvox you looked at, the paramecia that you looked at, the euglena that you looked at, those are all protozoans. None of them causes the silkworm disease, but that's who we're talking about here, right? So he connected a microorganism to a disease. And it might sound strange that we're throwing somebody up here, right, in the golden age of microbiology, 5 things, and one of those things happens to be a disease of a silkworm. And you think to yourself, why do I care about worms? Well, if you think back to the 1800s, right, if you've ever taken history classes, you learn about the Silk Road, right? The Silk Road was a major trade route between the East and West. And it's called the Silk Road because silk was a major traded commodity, right? So silk was the basis of national economies. So when there was no silk, because a protozoan was killing the silkworms, then people were starving to death because they didn't have anything to trade, right? So sometimes it seems weird that we study animal diseases because we think, right? I mean, he also studied avian cholera. You're like, who cares, right? They're chickens. But I mean, maybe you have a pet chicken and you do care, right? I told you I have a dog. I love him. People that study dogs, I love them too, right? But sometimes, right, the reason we study animals is because they have a direct consequence for human beings, right? In this case, we have a big monetary consequence when the silk trade is carbolixed by disease. So he connected those, right? And we have to remember, because we go, well, nadoy, things cause disease, right? But at this point in time, people thought bad air caused disease, right? There was something called the miasma theory of disease. So people thought, no, it just kind of happens, right? And we don't really know why. And when we don't know why something happens, it's really hard to keep it from happening, right? But when you learn why something happens, now you're armed and you can stop that thing from happening. So making this connection, right, this was one of the first times that a real clear connection was made between some kind of microorganism and a disease process. So not only was it helpful because it helped to protect the silkworms and keep that silk trade going, it also was really useful because it added to the general knowledge, right? Because if protozoans can cause disease in silkworms, maybe protozoans can also cause disease in human beings, which they can, right? There's a little fun thing called hiker's diarrhea, and it's pretty common even in the United States if you drink contaminated water, like when you're hiking, and it's caused by Giardia, which is a protozoan, right? So it's important on its face. It's also important because it adds to the body of knowledge, and then people go, I wonder what else protozoans do, right? So Louis Pasteur. All right, very quickly after that, 1876, we have a German gentleman by the name of Robert Koch, and he, super cool guy, father of epidemiology, came up with something that is called Koch's postulates. We're going to talk a lot about them in our epidemiology unit. Koch's postulates, right? Because Pasteur and other people published their data, right? Because this is a big thing during the Enlightenment. We learn, we tell people, and we allow them to scrutinize what we know, right? So that we have peer review. So Pasteur publishes his data, and other people publish their data. Jenner published his data on smallpox and cowpox, and Robert Koch goes, you know, what would be really cool is if we could figure out just a set of criteria to say this thing causes that disease. And he did. He came up with Koch's postulates, and Koch's postulates are still used today, right? Not as often because most diseases we know. But still today, when we have a disease process, we can use Koch's postulates to scientifically confirm that an etiologic or causative agent is causing that disease, right? So he's the father of epidemiology, right? So Edward Jenner, sort of the father of immunization, Robert Koch, the father of epidemiology, and so right here, we're just basically mentioning them. Later on, we'll talk more about these guys. And then again, right after this, right, so we're really starting to hone in on bacteriology. A guy by the name of Hans Christian Graham, sort of like Hans Christian Andersen, but different last name, right? This guy discovered the Gram stain procedure, which to anybody outside of microbiology, people are like, snooze alert, who cares, right? But you might have heard of gram-positive bacteria and gram-negative bacteria. And it turns out that understanding whether a bacterium is gram-positive or gram-negative is the first step, not only in identifying that bacterium, but treating it. Because we have antibiotics that work really well against gram-negatives, and we have antibiotics that work really well against gram-positives. So if you've ever given a sample to a lab, right, they swab you somewhere, right, with the exception of your stool, because we know there's going to be tons of stuff in there, one of the first things that happens is that that thing gets gram stained. And the lab calls the clinician and says they've got gram-positive cocci, or gram-negative cocci, or gram-positive bacilli, or gram-negative bacilli, and now the clinician knows what antibiotics they can start treating with if it's a serious infection. Now, unfortunately for Graham, he thought he was a total failure. Because what he was trying to do was come up with a way of taking lung tissue, right, taking a lung sample and looking for bacteria. So he was pouring, right, it was kind of a thing in the late 1800s. People were just like grabbing colored stuff and pouring it on cells to see what would happen. And what he was trying to do was find a stain that would make bacteria kind of pop from the lung cells so that they could be easily identified. And what he found was, when he did this procedure, a lot of times the bacteria just went away. You couldn't tell the difference between them and the rest of the stuff, and he's like, oh well, I'm a failure. Maybe someone someday will be able to figure out some useful way to use this staining procedure, and today it is the most commonly used staining procedure in the whole world of microbiology. So he may not have known that, but luckily he actually told people about it, and someone was like, wait a second, we can maybe use that, right? So they did. So that's our golden age, our historical golden age of microbiology, right? So this isn't a history class, one sec. So I don't want you to be like, okay, in 1880 whatever, this happened, right? What I would like you to do is think about, I mean, we do want to give a nod to these people, right? So I do want you to understand what each person did to contribute, but what I would like you to do in terms of timeline is think about these things in a series. What happened first? How did what happened first maybe lead to what happened next, right? Because if Jenner hadn't published his data, would Pasteur have thought to do the things that Pasteur did? So I want you to think, right, because knowledge is cumulative. And so I want you to think about these things in a timeline so that you understand that the things that come before, right, inform the things that come later. And what's your question?
For the Gram stain procedure, what was it actually testing for? Like what's positive and negative?
So we're going to find out in our next chapter that what it actually does is when you apply a stain to bacteria, some of them have a really thick layer of a substance called peptidoglycan, and so you can't pull the stain out of them very easily, and so they look purple. And then some of them which have a really thin layer, which he didn't realize, right, that's why they were decolorizing, is because that thin layer allowed the stain to come out. So when we do a Gram stain, which you're going to be doing Thursday, a week from today, we're going to be looking at that procedure, and it's a procedure that will employ multiple dyes and a decolorizing step, and at the end of that thing, some of them retain the stain that we use, and some of them don't, and so we call them gram-positive and gram-negative.
Why do they need different antibiotics?
Primarily, it's because those that have that thick peptidoglycan layer, there are actually antibiotics that target that peptidoglycan layer, so if you don't have it, then it's not going to be a target for you. And as we look at cell structure and as we look at different things like that, we'll talk more about that and that'll probably make more sense, but that's kind of a simple way of explaining it, right? So if you lack structure, then this doesn't do anything, right? It's like shining the light into a blind person's eyes. It's not, not gonna shock them, right? Because they can't see. So we're thinking about this sequentially, right? Because knowledge is cumulative and what people know, right? And that was really part of the Enlightenment, was letting people know what you know, right? Having that common language, which wasn't English back then, right? But having that common language of Latin or Greek to speak to one another and share with the whole world what you learned, right? And that was not common prior to that. So these guys are publishing data and people are learning and they're going, ooh, I wonder if I took this and that and then did this and boom, right? And other stuff happened. And even Graham, right, even though he thought he was a failure, he was working on the back of people who were using other stains to do different things. And he thought, ooh, maybe I could do this thing with some stains. So, chronology, right? It's roughly a 200-year period. Each person comes in a sequence. So the sequence and then what each person contributed, that's what I want you to focus on. But I'm not going to ask you on a test what happened in 1884 or 1876. I want you to think about the progression, right? All right, so fast forward 100 years. A team of scientists, Kohler and Milstein, fused cancerous cells with antibody-producing cells to make monoclonal antibodies. And maybe you look at that sentence and you go, okay, there's like 70% of that sentence, zero idea what it means, right? Like, okay, antibody, maybe you heard of that. Monoclonal antibody, what, right? So don't stress. When we get to our immunology unit, we're going to be talking about monoclonal antibodies. So I just want to give a nod to these guys and say, hey, like if there were no team of Kohler and Milstein, there would be no immunological tests, right? There would be no pregnancy tests because those rely on monoclonal antibodies. There'd be no rapid strep tests. There'd be no rapid staph tests. There'd be very few ways of diagnosing illness if it weren't for these guys coming up with this, right? So we're just going to say, there we go, Kohler and Milstein, right? And then we're going to fast forward just a little bit to a team of scientists led by Barré, Sinoussi, and Montagnier, who discovered the etiologic agent of AIDS in 1983. And though we haven't cured AIDS, right, we have gotten a handle on the transmission of AIDS. We're going to talk a lot about AIDS at the very end of the class. It makes a really nice wrap-up session to our immunology unit because it's a disease of the immune system, so we can see how dysfunction in the immune system leads to disease, right? And it's a virus that attacks the immune system, so we're going to learn lots of things. And one of the things we'll learn is that though, in 1983 we had a pandemic, and it did increase a lot from 1983 to maybe like 2010, but in the last 10 years, the total number of HIV cases in the world is leveling off. And one of the big things that led to the treatment and the blocking of transmission of that disease, right, was identifying what was actually causing it, right? Because in the United States, they discovered that people had this disease. It took them 5 years to figure out what was making people sick. So for 5 years, you can't stop it from being transmitted because you don't even know what it is, you don't know where it lives or what it does. So discovery of that etiologic or causative agent, right, was the first step in stopping the transmission of HIV, right? Unfortunately, right, when it was discovered, they said, we'll have this thing cured in 5 years. And that's not happened, right? That would have been 1988. And unless you've been in a coma, you know that this was not cured in 1988, right? But it still was helpful. So that's our golden age, right? So these guys in 1983, Kohler and Milstein in 70, what did I say, 75, right? Those are just, oh, that's cool. We're going to hear about them later on, right? So now the big thing, right? The big question. Metaphysically, microbiologically, right? Where did you come from? That's everybody's big philosophical question, right? So about 2,500 years ago, there was this super smart dude living in Greece, and his name was Aristotle, right? You might have heard of him, right? Pals with Socrates, Plato, all those guys, right? My daughter has to take classes. She actually read him. She's like, sucks. I hate you and I hate him, right? But super smart dude. And one of the things that he came up with that we go, really, was he that smart? Was this idea of what was coined Aristotelian abiogenesis. So Aristotelian, because he is Aristotle. Abiogenesis, because what he said was that non-living things can spontaneously generate into living things. And you're like, that seems super dumb, right? But, right, he's Aristotle. And he says that one of the things that can spontaneously generate is aphids. You know aphids, they're the little tiny green guys. They like to grow on rose plants, right? And he said aphids spontaneously generate from the dew that falls on plants, which sounds magical and beautiful, right? You're like, oh, the dew just becomes aphids. And he said flies spontaneously generate from putrid matter, right? Rotting stuff. Mice from dirty hay, crocodiles from rotting logs and water, right? So he's kind of running the gamut. Crocodiles are big, aphids are tiny. All of those things can spontaneously generate, right? And we look at that and we go, obviously an idiot. But we have to pretend that we don't know the things that we know in the 21st century, right? Because pretty much everything you don't know experientially. I mean, you might know not to touch a hot stove experientially because you have physically touched a hot stove and burned yourself, and now you know not to do that. Or you know to wear sunblock because you get a sunburn if you don't. But most of what you know is not due to your experience, it's because someone has shared that information with you, and this is 384 BC, right? So take 2019, add 384 to it. That's how long ago this was happening. No microscopes, none of any of those things we just talked about in the golden age of microbiology, right? So we got to cut him some slack, because all he's doing is observing what he can observe and making conclusions based on those observations. And when you don't have a microscope, you don't know that aphids lay microscopic eggs, and those microscopic eggs grow into aphids. And you don't know that flies lay little tiny eggs that are hard to see, right? So we don't know this stuff. So he publishes this, and it might be shocking to think, right, that until the 1600s, everybody just took that as fact. And that doesn't mean the dummies like living out in, the yokels out in the middle of nowhere that don't talk to anybody. That means that was the prevailing scientific theory. That these things, living things, spontaneously generate from non-living matter. So in 1646, Sir Thomas Browne is looking at this stuff, right? It's the Enlightenment. People are being encouraged to go back and really think about the things that they take as true. And in science, right, that means we got to think about things that have been taken as scientific fact, but there's never been any scientific study to determine whether there's a connection between rotting meat and maggots, right? So he publishes the Pseudodoxia Epidemica, where he says, here's all the stuff that people think is true that is dumb and not true. That's basically, right? Very many received tenets and commonly presumed truths, right? These are all the things that everybody believes are true, and they're not. I don't think they are. So this guy, Alexander Ross, responds, right? These are both scientists. Because you're like, well, I would side with Brown, right? I'm going to go with Brown. He sounds like a smart dude. Well, he puts this, and Ross responds, to question spontaneous generation is to question reason, sense, and experiment. If he doubts of this, let him go to Egypt, and there he will find, right, this is not a typo, that's how they spelled find back then, the field swarming with mice begot of the mud of Nilus to the great calamity of the inhabitants. So he's basically like, you're a total idiot. And anybody that thinks like you do is a total idiot, right? So 1646, I said you don't have to remember dates, but think 1668, Francesco Redi reads this and goes, challenge accepted. You think he's a dummy? I think you're a dummy. And he goes back and he looks at Aristotle's original declarations. And one of them is this idea that flies arise from putrid matter, right? Rotten stuff. And he says, I am going to test that theory scientifically. And he begins this debate between biogenesis, which he is a proponent of, which Brown is a proponent of, and spontaneous generation, which Aristotle was a proponent of. So, is it that non-living things can turn into living things, or is it that living things can only come from living things? Now, if you've had physiology or if you had your general biology recently, you might have learned of something called the cell theory, right? All cells are surrounded by a plasma membrane, and one of the things the cell theory says is cells come from pre-existing cells. But this, right, is what that rests on. Because what that says is non-living stuff can't become living stuff. It's not possible. That was his declaration, right? So he sets up a little experiment, right, pictured here. So he puts some meat in containers because he's been observing, and what he notices is when you leave meat out to rot, flies land on it. I wonder what they're doing. So he says, when they land on it, what I notice is then all of a sudden there's maggots there. So he takes some of his containers of meat and he covers them with gauze. Covers some with gauze, leaves some open. To us, it's no surprise that those he covers with gauze don't have any maggots growing in them. Those he doesn't cover have maggots growing in them, right? And he's probably feeling really good about himself. I just challenged Aristotle and I won. So he publishes his data, right? And we publish data so that the scientific community can scrutinize our data, right? Look at the conclusions that you've come to and see if the data really support the conclusion, right? And he's like, no problem, and everybody goes, yeah, no, you're wrong. Maybe maggots don't spontaneously generate because maggots are super complicated, right? I mean, they turn into flies. But animalcules, those little animalcules, those little weird things that we see swimming around inside of a microscope, they look pretty boring, right? They're just some blob that swims around. That thing, that simple thing, that can spontaneously generate. So maybe they can't, but we're still going to bet on Aristotle and not you, Francesco, because Aristotle, right, could explain how these animalcules arise. So he's sort of, it's like the Charlie Brown, when he's like the little red-haired girl and he can't talk to her because he's nervous, and he puts his head down and he walks away, right? So that's sort of Francesco Redi. And kind of just to just twist the knife a little bit, this guy John Needham reads about this study, and he's like, idiot. I'm so sure that animalcules can spontaneously generate that I am going to design an experiment to prove that animalcules spontaneously generate. So I am going to take some broth, and I'm going to boil it, and then I'm going to leave it sitting out with a cover on it, and lo and behold, animalcules spontaneously generated in the broth. So he publishes his data. He's feeling pretty good, but you know how your mom said, don't say nasty things about people, right? Because then you just invite scrutiny on yourself, right? So people turn the guns at John Needham, and they're like, listen, bud, I mean, you did this like sort of weenie boiling thing, really not that long, and then you left it sitting out on the countertop before you covered it. Like, no, it's not spontaneous generation. It was probably not sterile, and then when you left it sitting out, stuff fell in, animalcules fell in from the air, and boom, it's not spontaneous generation. So he thinks he's super smart. He publishes his stuff, and everyone's like, negative, dude, bad experimental design. Which is why it's super important not to just look at someone's conclusion or even just to look at their data, but actually look at the experiment that led to those data, right? Because in this case, if you just looked at the conclusion, you go, it's covered, I see a picture in a magazine and it's covered and it's full of bacteria and animalcules. This guy must know what he's doing because, he's a doctor. He's got a PhD, right? Some of the dumbest people I've ever met in my life have PhDs. Have you ever done that? You think you're like, how did that happen? So, debate is not over. Now, one of the guys who is in Francesco Redi's camp says, oh, John, let me just show you how wrong you were. I will do the same thing you did, but I will do it with proper experimental design. And when I do it properly, no spontaneous generation, right? I will boil some and leave it open. There's your animal kills, John. And I will boil some while it's closed and leave that, and nothing will grow, and boom, right? Here I am. My name is Lazara Spallanzani, and yes, I have solved this question. I have answered the debate, and there is no such thing as spontaneous generation. Fortunately for Spallanzani, people look at his experimental design, right, and they go, I don't really like that you left that cork on there the whole time, because at the same time this was done, somebody had discovered something they called the vital force. Now, we don't call it the vital force anymore, we call it oxygen. And what they learned was, when you take things and you seal them in a container and you don't give them vital force, they die, right? Which you could, I would not suggest that you learn that experimentally, but you could, right? Many young children try to learn that experimentally, and of course, as soon as they pass out, if they really have the chutzpah to do it, they pass out and they start breathing again, right? But the vital force was discovered. They didn't know what it was, but they knew that organisms need this vital force to live. And they said, well, you kept it out. So how could they spontaneously generate if they don't have a vital force, right? So he thinks he's a smart guy, and they say, nope, right? They need the vital force. That's why they didn't spontaneously generate. And again, we're back to square one. Aristotle is the king, and people are trying to figure it out, right? So, because knowledge is cumulative, right? A guy by the name of Theodore Schwann, those of you who have taken physiology, you had to look at Schwann cells, same dude. Theodore Schwann decides he's going to throw his hat in the ring. He looks at all this stuff, and he goes, piece of cake. I just don't use a cork. Easy enough, right? And I don't know what's going on. Like, I like to think about what is going through the minds of people when they're doing things. So what I think is, Schwann was like, I'm going to leave my broth, I'm going to sterilize it and leave it open, but I'm a little bit nervous. I'm a little bit nervous about just leaving it open. So I'm going to take this little flame and put it there, just in case. Right? So this is open. Vital force can get in. Broths are sterilized. They sit there. Nothing grows. And he's like, sweet, I did it. But he did it, right? Because people were like, listen, Theo, the vital force, maybe it doesn't like to get hot. Maybe you've damaged it, right? So they said, no, the vital force was damaged by the heat. So here's the bummer, right? He actually solved it. But he didn't trust it, and so he made it more complicated than he had to. Did you ever do that with something? Super easy explanation, you make it more complicated, and then something goes wrong, right? So that is what Schwann did. Too complicated. So a guy by the name of Louis Pasteur looks at all of this, and Louis Pasteur, he's a smart dude, right? He knows to look and see what other people have done. I mean, he's a smart dude in his own right, like not that I'm taking it away from him, but he is a very good at learning from other people and figuring out how to do it right, which is a great person to have, right? So he looks at this and he's like, oh, Teddy, all we have to do is get rid of that little flame and everything will be fine. So he does. Louis Pasteur designs this cool flask with a swan neck. Right, a swan neck. So you picture a swan, right? Swans have really long necks, right? So he has this really long neck, and he heats it so that it curves. And then he takes his broth, and he boils it in the swan-necked flask, sets it on the countertop in 1864, and if you go to the Pasteur Institute in Paris, it is still sterile today. It's open to the air. The vital force has not been damaged because it's been room temperature for almost 200 years. And just so that nobody could say anything about it, he did multiple swan-necked flasks, right? Because every experiment always done with more than one experimental piece. He takes half of them and he breaks the swan neck off and leaves those out. And guess what happens? Animal kills. So he proves, even though prove is a dirty word in science, right? We don't prove, we disprove. Everybody always says prove and I cringe. It's like when they say data is and I go, oh, it's a plural word, right? You don't, I know, but I do. So he shows that this is an environment that is capable of supporting animal kill growth by breaking the swan neck and leaving it open, and animal kills grow there. But he also shows that if you don't allow microbes, right, animal kills to fall in from the air, then nothing grows. And finally, after 200 years, this debate between biogenesis and Aristotelian abiogenesis, or spontaneous generation, is over, and biogenesis wins. One of our instructors made this a few years ago. Now, you are not enough of a microbiologist perhaps yet to understand that when you see broth and it's clear like this, there's nothing in it. And when stuff grows in it, it becomes turbid or cloudy. But you will, because today you're going to be inoculating sterile media with microorganisms, and the way that you will initially tell whether they've grown is because that broth will be turbid when you take it out on Tuesday. But this, a couple of years ago, was done. Here's his swan neck, right? It's open, but nothing is growing here, because any animal kills in the air are getting trapped right there, right? And they can't climb up the glass and fall inside. He also had another one that just had a straight top on it, so it was open right here. But we had to get rid of it because everything grew inside it, and we couldn't have it anymore because it became biohazard, right? Because it had a bunch of muck growing around in it. So this is a more modern example of the swan neck flask that Louis Pasteur satisfied all of the demands of the scientific rigor about the biogenesis debate and finally laid it to rest, right? So since the 1800s, we have known scientifically what you probably knew in your gut already, and that is that non-living stuff, right, it doesn't spontaneously generate into living stuff. So no, I'm not going to answer the philosophical, right, where did you come from? But I can tell you microscopically, microbiologically, right, what does science say cells come from? Other cells, that's where the cells come from. So that's the biggie, right, is that he formulates this conclusion that biogenesis is supported, spontaneous generation is disproved, and now we're done with chapter 1. So now, if there's something that you're like, ooh, I didn't really understand that very well, right? You've got the chapter 1 outline. Once you've got everything dialed in, you've got your chapter 1 quiz that you can take online, right? I'm not going to check them. I'm not going to know that you got a question wrong and like put a little mark in my book that you're a dummy, right? But if you want to do those things, we're done with chapter 1 and you can do those things for chapter 1. And now we're going to move forward to chapter 4, right? So we're completely going over chapter 2. We're not going to go into chapter 2. We're bypassing chapter 3 for right now. Chapter 3 is really just a chapter about microscopy, so that's why we talked a little bit about it on Tuesday, because we used our microscopes for the first time on Tuesday. And we will talk a little bit about it today, because we're going to talk about, what were we talking about it today? Actually, I think we're going to just stop at chapter 4 today. All right, but we'll talk again about chapter 3 on Tuesday, because we're going to be doing staining of microorganisms on Tuesday, and so we want to have a little bit of background for that. So we're going to jump forward to chapter 4. Chapter 4 deals with cells, right, which is a nice segue, because we just talked about where do cells come from. Cells come from pre-existing cells. So what's a cell? My son asked me last night at the dinner table, because my daughter says, other people don't have conversations like this. She goes, I go to my friend's house. They don't talk about diseases or like weird stuff, but we do. And he goes, what's the difference between a bacterium and a virus, right? So one of the things, I said, well, bacteria are cells. So we're going to talk about cells. But this is microbiology, not general biology. And in general biology, nobody cares about the bacteria. They're like, oh, the prokaryotic cell, it just doesn't have a nucleus, right? And that's all they say about it. And then you talk about the nucleus and how amazing the nucleus is because it protects the DNA. But my question to you is, if it's so important, why don't prokaryotes have one? Then you talk about endoplasmic reticulum and Golgi bodies and vesicles and all of those things, right, which we would call together membrane-bound organelles. So since you've already done that, we're not going to do that here. What we are going to do in chapter 4 is give due to the bacteria. We are going to talk about the prokaryotic cell, and we are going to talk about those things that are specific to the prokaryotic cell. Because you've already learned about cell membranes, because you did that in general biology, right? And it's not our job to go re-talk about all the stuff that you already know. It's our job to add more to it, right? So we're going to focus on the prokaryotic cell. And initially, right, we're just going to look at them. So here's a prokaryotic cell who has lots and lots of things, right? So he's not just some dude who's tiny and doesn't have a nucleus. Those things are both true. They are small and they lack nuclei, but there's a lot going on, right? So we see a cell, right? We know he's a cell because he's bound by a cell membrane. That cell membrane, by the way, lipid bilayer membrane, just like everybody else's, right? Nearly indistinguishable from your own lipid bilayer membrane. Looks the same. Got some DNA, got some ribosomes, so we're really not going to talk about those because those are things that you talk about in general biology. Has lots of other things that we don't find in eukaryotic cells, so that's where we're going to spend our time. And the way that we're going to do that is going outside in. So we'll start really with things that aren't even on the outside of the cell. They're actually outside, right? How do we know they're outside? Because they're outside the plasma membrane, right? So we're going to start on the outside. We're going to take like a microscopic view, right? We're going to start far away and we're going to come closer. And when we start really far away from microorganisms that are prokaryotic, what we notice initially is that unlike eukaryotes, they typically have a defined shape, right? So you don't have to raise your hands. But you can think about if you've been in an anatomy class and you had to identify all the different kinds of human cells, and you're like, right, they show you a picture, nothing ever looks like the picture. You want to know why? Because there's no cell wall. And so animal cells change shape all the time. Well, the nice thing about bacteria is that they have a cell wall which is rigid, and so it gives them a shape. And we can actually identify them. One of our first identifiers for bacteria is what their shape is. You are doing a lab today with E. coli, Escherichia coli. All right, no common name. You can call him E. coli if you want to. That's sort of his nickname. It's the only nickname he's got. He is a single bacillus. He's always a bacillus because he has a cell wall that makes that rigid structure, and he's never anything other than a bacillus. And he is genetically predisposed to separating completely when he makes baby E. coli, and so when you see him, he always is single bacilli. Which means if you see single bacilli, you could be looking at E. coli, which is something you're actually going to be checking on Tuesday. You're going to try today to do one of the most important techniques in all of microbiology, and that is aseptic technique. Transferring what you want without transferring things you don't, right? Aseptic, without contamination, without growth of other things. The way that we will test that is not super rigorous, but we don't want to spend the whole semester testing it, right? Is we are going to look at what you have done under a microscope and look for single bacilli. And if we see single bacilli, we will say, well, we know E. coli are always single bacilli, so if I see single bacilli, there's a good chance that that's E. coli, because that's what you actually took and put in there. If you see cocci, round bacteria, then you contaminated your E. coli, because now you have something that can't be E. coli. Now, does that mean every single bacillus on the whole planet is E. coli? Nope. But it means if it's a single bacillus, it could be E. coli. So, the two most common shapes for bacteria, and the two that you need to be able to recognize microscopically and communicate with appropriate microbiological terminology, right? We don't say round and rod-shaped, we say cocci and bacilli. So cocci, right, Latin, bummer of a language for those of us who speak English, right? Because what do we put on the end of words to say that they're plural? An S. Guess what they don't do in Latin? Put an S at the end of words. Actually, words that end in S are singular. So coccus is the singular of cocci. And that is bacteria who are round. If I bebop all the way over here, bacillus is the term that we use to describe rod-shaped bacteria, right? They look like a loaf of bread. So bacillus, bacilli, coccus, cocci. Now we have a couple of others, right? Coccobacillus, right? So the name sort of suggests what's happening. You took the word coccus and you took the word bacillus and you squished them together, and that's kind of what you have when you see a coccobacillus. Is this little guy he's small and he just looks like a bacillus that someone went and sort of squished him. Now here's the reason we don't really focus on him. With your microscopes, when you make them 1,000 times their size, you can't tell that you have a coccobacillus. You would see it and think it's a cocci. So we have cocci microscopically and we have bacilli microscopically, right? There are other shapes, right? Vibrio is sort of a curved bacillus. Spirilla, whose singular is spirillum, is sort of a spiral, which is where it gets its name. And spirochete is a corkscrew-shaped bacterium. Now, like I said, the ones that we're going to pay most attention to are the cocci and bacilli, and you want to get used to using those words, right? When you're looking at things under the microscope, you want to get used to saying, I see single cocci or single bacilli. Or maybe you see what looks like two cocci stuck together. And if you see that, you will say, I see diplococci. Diplo means two. You can also see in your microscope diplobacilli. Now it's important to understand when we see diplococci or diplobacilli, it doesn't mean that they're crowded, right? It's not like this, like you're just touching. I'm going to touch you, I'm sorry, right? We're not conjoined twins. We're just next to one another, right? These cells are separate cells. They have their own plasma membranes, but when they divide, they don't detach from one another all the way. And the cool thing about that is, like everything else about a cell, it's genetically predetermined. Everything a cell does is based on its genetics, right? Genes get transcribed and translated into proteins, and proteins do jobs. In E. coli, one of the jobs of the cell when it replicates, when it makes a baby, is for the cell membrane and the cell wall to completely separate and the cells to become detached from one another. Now, because that's genetic, when we see single bacilli, right, that means we can tell something about the genes of that cell because the genes told it to do that. When we see diplobacilli, we know that the genes in that organism told it to do something different. It said, separate your plasma membrane, but don't separate your cell wall and stick together. So when we see these different arrangements of cells, that can be our first step in identification. And it's more so, it's not really a positive identification, but it rules things out, right? Like if I see diplobacilli, that's not E. coli, because E. coli is never diplobacilli. If I see single bacilli, it could be E. coli, because E. coli is one of thousands of single bacilli. Chances are, if you in a lab have been handed E. coli and you put something into a test tube and you look at it and it's single bacilli, chances are it's E. coli. Because out of all the thousands of things it could be, what's the most likely explanation? That it's the E. coli. So morphology, which refers to the shape of bacteria, and arrangement, which refers to how they're put together, are really important diagnostic tools because they're little, we can't see inside them, can't ask them questions. So observing what they look like microscopically is our first step in identifying them. Yes?
For the one that ends in E2E, how is that made plural?
Spirochetes. It's the only one that gets an S on the end of it. I'm not going to yell at you if you say cocci instead of coccus. It's okay, right? But we don't want to say round. We want to say cocci. And we don't want to say rod-shaped. We want to say bacilli. Even though a lot of times you'll hear rod-shaped, right? That's a more common thing. You'll never ever hear anybody say circle-shaped. But you might hear people say rod-shaped. Right? So notice the rest of these have only single arrangements. Right? Single spirilla, single vibrio, single cocobacilli, single spirochetes, right? It's only the cocci and bacilli that can be arranged in diplo. And it's only cocci and bacilli who can continue that concept of dividing into two cells but not detaching from one another and grow into chains. And chains are strepto. Streptococci, streptobacilli, right? So Streptococcus pyogenes, which we talked about on Tuesday, is named Streptococcus pyogenes, right? Because there was a pussy wound. So they said, oh, we're going to call him pyogenic. And when they looked at that, the bacteria from that pussy wound under the microscope, what they saw, they said, ooh, I see Streptococci. Hey, let's call it Streptococcus. And that's why it's really important, right? If you're talking about the genus, you got to capitalize it and italicize it, because you otherwise could just be describing streptococcal bacteria, right? Because it's just an adjective. And this is where the adjective comes from, right? Words that mean chains of round cells. Streptobacilli, chains of rod-shaped cells. Now, notice the cocci can get even more complicated, right? Yeah, question.
So when it's streptococci, is it also touching? I mean, is it also like?
So what it would look like if you could look microscopically, right? So here would be all of their plasma membranes, right? So they are single distinct cells. But if you looked at their cell wall, they would kind of look like that. Like they would be touched and attached to one another.
Okay.
So the cells are distinct, but the cell wall sticks. So it's not a matter of being crowded, it's a matter of actually being attached to one another physically. But, right, so like conjoined twins, but unlike conjoined twins, they are separate, right? They don't share anything. They're just attached to one another. Now, cocci, because of the way that they reproduce, can undergo a cell division on more than one plane, right? Bacilli can only undergo division. They get longer and then they cut in the middle, right? And they either sever from one another or they stay attached. Cocci can do that, but they can also go that way, which means that you can have a group that goes out this way and goes out this way and forms what we call a tetrad. Now there are some cocci that get super crazy and they go like this, and they go like this, and they go like this, and they make a cluster. And a cluster, in Greek, is staphylo. Staphylococci means a cluster of cocci. And there's another very famous genus of bacteria, Staphylococcus, which has Staphylococcus aureus, Staphylococcus epidermidis, all sorts of stuff, right? And the reason that it is like Staph aureus, aureus means gold, right? Just like if you look on the periodic table, what's the symbol for gold? AU, right? Aureus means gold. So when they found it on a petri dish, they said, oh, it's gold colored. And when they looked at it under the microscope, they said, oh, it's clusters of cocci. Let's call it Staphylococcus aureus. So it's an adjective, but it's also a genus name, right? Which again tells us why it's so important that when we're talking about a particular genus, we capitalize and we italicize so that everybody knows what we're saying. So, tetrads could go into these funky things called sarcinae, staphylo. What we really pay attention to is single, diplo, strepto, staphylo. Notice staphylo is only possible with cocci. There's no such thing as staphylo anything else. You could have, right, when you look at your E. coli, especially when you get them from your agar slants, right? Because that's a solid media, and bacteria usually grow faster on solid media. And it's easier to grab a bunch of them out of solid media. So you'll get a visible chunk of organism, and you'll put it on a microscope slide, You'll smear around to try to spread them out. You'll stain them with your simple stain, and then you'll put them under the microscope, and there will be a spot on that slide where they're all crowded. And you go, oh, they look like Staphylococcus. I am here to tell you that is not possible. That's crowded. That's like if I took everyone in this room and I crammed you into the corner, that does not mean that you're all one single multi-personed person. It means you're crowded. So we move away from, right, and the crowded area is good. It's nice because they're easier to find when they're crowded, right, because there are bunches of them. But we move away from where they're crowded, and we look in the area where there aren't so many of them, and that's where we can establish the true morphology and the true arrangement of cells, right? So these, none of these are light micrographs. And you could have figured that out. Or if you think about it, you go, well, I mean, here's a 1 micron bar, right? You know what 1 micrometer is now. That was 10 of them fit in the space between your 2 tiny tick marks. So do you think you could get this kind of magnification with a light microscope? Negative. So these are all electron micrographs. And they're also all scanning electron micrographs because they're trying to show you the 3-dimensional structure of these cells, right? So that you can see how they're attached to one another. We don't want to see through them. We will see some things, right? In fact, if I just flipped back here, right, that is a transmission electron micrograph because they want to show you the DNA inside of the cell. These are scanning electron micrographs because they're trying to show you the surface, right, the shape of the surface. So you want to start practicing and be able to recognize cocci and bacilli and strepto and staphylo and diplo and single arrangements. And the way that you'll practice that, right, is next week we're looking at bacteria under the microscope, lots of different bacteria, and you are going to start describing them using those words, right? And if you don't, I'm going to say, so what would you call that, right? What's the fancy way of describing that? You will say, oh yeah, cocci, right? We get used to that. So, right, so we're way outside, right? We're using our 4x objective, right? We're zooming in. We're using our 1000x objective. Now we can see the shape, not to this extent, right? Because this is way more than 1,000 times their size. But we can see the shape at 1,000 times their size. And then we get a little closer, right? Meaning closer than we can really see with our light microscopes in order to see what's going on the surface of the cells, right? So now we're going to zoom a little bit closer, and we're going to look at the cell, but we're not going to go inside yet. We're going to look at the outside, because it turns out there are 4 major structures that prokaryotes could have, not that they all have, but that they could have, that are external to the cell because they are external to the cell membrane, that lipid bilayer membrane, right? The first of those is something called a glycocalyx. And a glycocalyx, right, if we took apart that word, it really just says slimy polysaccharide, and that's what it is. It's just goop. That goop, though, it turns out, is not just goop, right? And this is where having microscopes and having electron microscopes and all of those really high-powered microscopes is really important, because what just looks like goop to you and I, it turns out, can actually exist as goop, which we call a slime layer, or it can be really highly organized into something called a capsule. So these bacteria here, transmission electron micrograph, right? Because we got to get really close in there. And they've been colored, right? Because of course electrons, they don't make light, so they come out black and white, and you just color them to make structures more obvious. The cells are this blue thing. The capsule is this sort of aqua thing on the outside of it. It turns out that a capsule is a really nice thing for bacteria to have, but it's not so nice for you. It makes them highly pathogenic because it allows them to attach to your body, which we are going to learn when we go into our immune system, that the first step in disease is attachment. They got to attach to you, right? If you think about it, you've got all these things to try to stop them from attaching, right? You sneeze, you cough, you get diarrhea, you vomit. All of those things are your body saying, get off of me. And they're like, haha, no, I have a capsule, I'm not going anywhere. And then if they get inside your body, you have these macrophages and these other phagocytic cells that come over And they're like, haha, and they eat them, right? That's your first defense, is for you to eat those cells, right? These bacteria come in, they want to make you sick. The macrophage or the dendritic cell or whoever this phagocyte is comes in and he's like, I don't think so, grabs him, eats him. Well, the problem is when you have a capsule, it's really hard for that cell to grab onto you. You slip right out of their little pods, right? Sort of like trying to grab a water weenie. You know, I got one, right? Those little water weenies, right? You try to grab them and they slip out of your hands, right? That's sort of like a capsulated bacteria. The phagocyte tries to grab him, but because he doesn't have two hands, he's trying to grab with one, it falls away. And we know that capsules are really important because in the 1920s, a big study was done, and when they injected encapsulated bacteria into mice, they died. And when they took the same bacteria with no capsule, the mice were totally fine. What's the difference? The capsule. The capsule is a really nice thing for bacteria to have because it helps them to attach to where they want to be, and it helps them to prevent phagocytosis, being eaten by your immune system cells. So, the capsule and the slime layer are both kinds of this glycocalyx, right? So that's on the outside. Second thing on the outside, a cute little structure called a pilus, right? Which is again Latin, so the singular is pilus, the multiple is pili. So this is a pilus right here. Here's one cell, here's another cell. A pilus is a tunnel through which one cell can transfer DNA to another. So we will talk more about glycocalyces when we get to our body defense and immunology unit. We'll talk more about pili in our genetics unit because pili can be used to transfer DNA from one bacterial cell to another, right? And actually in this picture, I'll show you a close-up one, but this one, notice this guy looks kind of hairy, and he doesn't. Those hairs are our next external structure, the fimbriae. Fimbriae are just like bacterial hairs, and they're hairs, but they're kind of like bacterial Velcro, right? Velcro helps you stick to things, and fimbriae help you stick to things. So like the glycocalyx, the fimbriae can help them to attach to a substrate, right? And that substrate could be your nose, it could be your urethra, it could be your vents of your air conditioner, it could be the pipes in your sewage system, whatever that place is, right? They like to be somewhere, and this helps them stick in that place, right? So fimbriae are small hair-like structures. They might look like cilia, they're not cilia, right? Cilia, you'll remember from general biology, those are for motility, right? These are not for motility, they're rigid structures, they don't move, and they're just used for sticking. So think Velcro when you think fimbriae. And then finally we have flagella, and you're like, well, you said we were only going to talk about things that are specific to prokaryotes, and I know about flagella because we talk about flagella in general biology. Flagella are like gigantic cilia, and they're for motility. Totally true, but look at this. Whoop! That is one possible flagellar arrangement that exists only in prokaryotes. So prokaryotes have flagella. They use them for motility. They are absolutely different in structure, and they're absolutely different in the way that they are arranged on cells, right? For the most part, eukaryotes have a single flagellum, right? Think sperm cell. If you've ever seen a video of a sperm cell, if you've never seen a video of a sperm cell, I don't know what rock you've been living under, but maybe you've seen a tadpole, right? Tadpoles have a single tail, normally, right? And that single tail whips and it propels them forward, right? So that's how eukaryotic flagella work. Prokaryotic flagella are all different kinds of arrangements, and they move totally differently from eukaryotes. So even though they're both organelles of motility, we're going to talk about them because this is a very different kind of flagellum, right? So right off the bat, we have 4 different arrangements of flagella. And if you knew Greek, You'd have a lot easier time than this than if you don't know Greek, right? And again, we don't want to be angry with people that discovered these things. We want to remember they were trying to be nice to us when they gave them these names because everybody spoke Greek, right? It's all Greek to me didn't mean I don't know what that says. It meant, oh, I got it, right? So they looked at these things and they said they look like long hairs. Let's call them trichus, because guess what the Greek word for hair is? Trichus. So they looked at them and so maybe like a sperm, right? Well, they're like, well, that's one hair, so I'm going to call that monotrichus. One hair. Boom. Easy, right? And then they looked at another kind of cell and they were like, this dude has hair all over him. And they called that peritrichus. You actually know what peri means because when you were younger and you took math, you learned how to determine the perimeter. And what is the perimeter? It's the distance around. Peri means around. So peritrichus means around, everywhere. So those are our two extremes. You can have one, you can have them everywhere, and then in the middle you can have these lophotrichus where you have a tuft, several flagella at one end, because guess what lopho means? A tuft. So lophotrichus means a tuft of flagella at one end. And amphitrichus only exists in spirochetes. Amphi is another one that you actually, if you think about it, you know what this means, right? Think about going to a movie theater and going to an amphitheater. An amphitheater has seats on both sides. So amphi means the sides. See? It's not all Greek to you. Some of it you know, right? You just have to think about it. Amphitheater, seats on the sides. Normal theater, seats only in the front, right? So on the sides, flagella on the sides. So we have peritrichus around the whole area, monotrichus one, lophotrichus a tuft, and amphitrichus on either side. The other thing that's different about prokaryotic flagella is that they are rigid structures, right? Like a eukaryotic flagellum, it goes like this, right? You just have something at the end swinging a flexible structure. A bacterial flagellum is more like a corkscrew, right? I don't know, maybe you're not 21, you're like, I've never seen a corkscrew because I don't drink wine because I'm not 21. Pick a corkscrew, right? It turns. It's a rigid structure, right? And what do we do with a corkscrew? We turn it into a cork, right? And it burrows into the cork. That's how a flagellum works. It burrows through things, right? It works like the outboard motor of like a dinghy or the propeller of an airplane, right? It spins just like an airplane propeller spins and that moves air, or a boat propeller spins and that moves water, that propels them forward. Yes?
So for flagella, are they much longer than the fimbriae?
Yes. And fimbriae are stationary. Fimbriae never move. So if you had fimbriae on these cells, they would be just like maybe in this kind of dark area right here. I mean, there's not any pictured here, but they're much, much shorter. And they don't move, they're just static little sticky things, or hairy things, right? So when a bacterium has a single flagellum, it moves just like a boat. You move the propeller one way and the boat goes forward. If you want to change direction in a boat, you turn the direction of the propeller and the boat goes backwards. And that's exactly what these cells do. So you can watch them and they go, brrr, they just go, 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 and then they go, oh, I changed my mind, and they go like this, right? Because that flagellum is changing direction, and going this way makes them go forward, and going this way makes them go backward. Now imagine that you have flagella all over you, and they're all spinning at the same time. So you just kind of go, right, and you just fall all over the place. You tumble, right, and then you get them all together, and then you go forward, and then something happens, and you go, I changed my mind. And you change the direction of all of them at the same time, and you tumble. And so they actually do this thing, they call it run and tumble, where bacteria, you can watch them do it under the microscope. You're going to get to do this next week. They will run and then do these cute little somersaults. And they run and they somersault, and they run and they somersault. So you may not be able to see their flagella because these are transmission electron micrographs. That gives you an idea of how small flagella are. You can't see them, but if you see an organism just running and running back and running and running back, then you can infer from that that they have polar flagella because they're moving in these straight runs. If you see them run and tumble, you can infer from that that you are seeing bacteria who have peritrichous flagella. So you can actually infer that from looking at them under the microscope, even though you can't see that. So you're going to get the opportunity to do that. Now, these guys, I said, these amphitrichous arrangements are only found on spirochetes. And it's super cool because it actually will allow you to see what a flagellum looks like when it moves. Because these crazy suckers, it turns out they don't just have flagella outside, which is what everybody else on the planet has. They have flagella through their whole cell. So some books will say, oh, those are endoflagella, right? Because what does endo mean? Inside. And some other books, just to make your life difficult, will call them axial filaments, because they run along the axis of the cell. So you'll see both of those things, they mean the same thing. And what they mean is a flagellum that starts at one end and only one kind of cell, and that kind of cell is a spirochete, and it runs the length of the cell, and when it turns, the whole cell turns around it. And we're going to look at a video next week. I'm going to show you a video of bacteria swimming so that you can see that before you look for it in your microscope. And we'll get to see a spirochete, and the really cool thing, you'll see him, he'll look like a corkscrew, just right through the media, right? And then you can visualize, you can picture what that flagellum actually looks like, because something way bigger than it is stuck on the outside of it, moving in the same way. Now that corkscrew movement of that cell is actually a super huge bummer, because one very famous spirochete is Treponema pallidum. Hopefully that's a completely unknown word to you, especially because hopefully you've never had this, because this is the causative agent of syphilis. And how you get syphilis, right? Imagine that corkscrew going into the cork. Imagine the spirochete burrowing into your little nether regions, right? Because this is a sexually transmitted disease. So the chancre, the lesion that results from a syphilis infection, is actually because those syphilis bacteria got out of somebody's hoo-hoo and got onto your hoo-hoo and burrowed into your skin. But it doesn't hurt, right? The diagnostic for that is a painless necrotic lesion. If it's painful, it's probably herpes. But the cool thing, right, we don't use them in the class. Can you guess why? They're not biosafety level 1 or 2 organisms, right? We don't use them in this class. But we will look at a video because they allow us to see what that flagellum looks like. So when you want to see what a eukaryotic flagellum looks like, you can look at a sperm, right? Because his flagellum is so big we can see it with a light microscope. We can't see theirs with a light microscope, but what we can see is this spirochete, right? Because he has this flagellum, and his body is like this thicker version of that flagellum, and when he moves, right, you can see him because he's big enough to see with a light microscope, and he curves. He doesn't have a cell wall, by the way, so his shape changes with the shape of the flagellum. So it's kind of fun, right? You don't want to get syphilis, but it's fun to look at it under a microscope, right? Especially because you know what happens if you don't treat syphilis. You go cuckoo. You get a little rash in your hand, you're like, oh, that's not a big deal. And then 20 years later, right, you're woo, because you have syphilis eating your brain, which is super fun. I've been told, I don't know. So, flagella are used for motility. They're different in structure, they're different in arrangement, and they are one of the most important tools that a bacterial cell has to do what any cell wants to do, and that is to respond to environmental change, right? Because how do we know things are alive, right? What's the difference between a rock and a bacterial cell? A rock just sits there. It gets hot, the rock sits there. It gets cold, the rock sits there. It's dark out, the rock sits there. It's light out, it smells bad, it smells good, the rock just sits there. When those things happen to a bacterial cell, they will move. They recognize changes in the environment and they will move. They will not only move away from things they don't like, they will move toward things they do like. And the way that they do that, right, take a deep breath, don't freak out. I'm going to show you, right? This is a signal transduction pathway. Those of you that are physiology are like, no, not the G proteins, right? But basically, here's the thing about signal transduction pathways. You ever seen somebody line up dominoes, right? Dominoes are not supposed to be used this way, but how does everybody use dominoes? We line them up, and then we flick the first domino, and all the dominoes fall. And you watch it, right? And it's this exciting thing, watching the dominoes, and then something happens at the end, right? These signal transduction pathways, that's what it is. Something in the environment knocks over the first domino, the dominoes fall, and something happens as a change, and that's your response. So something comes, knocks down a domino, and the result is the flagellum changes direction. So I'm swimming, woo, that smells bad, and I swim backwards. So it allows me to respond to environmental changes. I don't like that, I want to get away from it. So we say that they are capable of responding to their environment. And one of the big ways that they do, if they have flagella, because not all bacteria do, is they will change the direction of their flagellum. They will recognize this chemical, right? It's either a good chemical, they like it, they're attracted to it, it's a bad one. It could even be light. Let's say it's a photosynthetic bacterium. Where does a photosynthetic organism want to be? In the dark or in the light? It's not a trick question, they want to be in the light, right? They want to move into the light. So they actually have photoreceptors, right? Like you have in the back of your eye, that's what your retina is, a photoreceptor. They have photoreceptors that can not only sense light, they can actually preferentially move toward the exact wavelength of light that they use for photosynthesis. So if you put an incandescent bulb, which you'd not be able to do in the state of California anymore because you can't buy them here anymore, and a fluorescent light bulb here, if you put them in the middle, they will swim toward the incandescent bulb because that's the kind of light that they photosynthesize in. If you scooted them over here, and you're like, well, but I put you right there, isn't that good enough? Negative, it's not. They're going to move away and they're going to go toward that. So it's not just that they will move out of the dark. They have receptors that can tell them, this is 472 nanometers. And they go, oh, I don't like that one. And they leave. I like 6 something, right? That's where I go. So they register some change in the environment, and they move in response to that change. So it's a big way that bacteria can change what's going on in their lives, right? But they don't do it like you do because you're big and they're little, right? You sense things spatially, by which I mean, if I am standing and half of my body is in the sun, and half of my body is in the shade, and I start feeling hot, I know that half of me is cooler than one half. And I will move my whole body into the shade because I can spatially separate something that's happening here to something that's happening here. My body's actually in 2 environments. But they're little, right? So they can't sense things spatially, right? They don't have eyes, so they can't go, God, I'm hot. Ooh, there's some shade over there. I'm going to walk toward that shade. They experience things temporally. They just move and see if it's better or worse. So really the only sense, right, it took me, I'm very proud of myself for thinking of this because it took me a long time to try to figure out, like, what do we have that's like, it's like smell, right? Your sense of smell is a chemical sense. Think about that the next time you smell something nasty, right? You smell rotting food, that's actually particles of rotting food landing in your nose, triggering a chemoreceptor or mechanoreceptor and sending a signal to your brain that that stinks, right? When you smell cookies, they go, ooh, that's nice, right? That's because molecules are coming from the oven out into the air, landing in your nose and triggering a mechanoreceptor, right? So that's how they sense everything, right? So imagine you go home today after class, right? You really like chocolate chip cookies. And you walk in the door and you're greeted by the glorious smell of chocolate chip cookies and a blinding light. So the only sense that you have available to you is your nose. So how do you find the cookies using your nose? You close your, I imagine closing your, I'm not gonna really close my eyes because that'll be embarrassing when I crash into something, right? But you walk toward the smell. And if the smell gets stronger, you continue in that direction. So you like it, you keep moving toward it. And you sense, ooh, it's stronger, stronger, stronger. Oh, wait a second, I don't smell it as much anymore. So what are you gonna do, right? Just rationally, logically. Are you gonna keep going in that direction? You're gonna turn. How do they turn? They tumble. So they move, and then they go, ooh, there it is again. There it is. Oh, I lost it, and they tumble. And so when they smell something they like, which is the only sense they have, they stop tumbling and they run toward it. And as soon as it starts to decrease, they tumble to try to get back there again. So they might look, when you look at them under the microscope, you might think that the running and the tumbling that they're doing is random. It is not random. They are directionally moving toward the things that they like, and they are tumbling for two reasons. One, because they lost the thing they like, so they're turning around looking for it, or, ooh, they just got into swift with something bad, right? You're looking for your cookies, and all of a sudden you walk into the bathroom, and you're like, well, that is not where the cookies are, right? So you turn around. So they run and they tumble in order to get to the good stuff. So they exhibit chemotaxis, taxi, right? Like what do you hail when you need to go somewhere? A taxi, right? Because it moves you. Taxis is movement. Chemotaxis is movement in response to a chemical. Phototaxis is movement in response to light. Now, if you don't photosynthesize, it doesn't make any sense for you to have a photoreceptor, but you will have chemoreceptors, and their surface is studded with different kinds of receptors, right? And they register glucose, they register lactose, right? They register good stuff that's yummy to eat. They register things like ammonia, which are poisons, and they swim away from those. So they have all of these different receptors on their surface, and it allows them to respond to environmental changes, to go where the good stuff is and get away from the small stuff. But they have to do that by blindly moving forward and hoping that things get better. And if not, they turn around and go back the other way. So you'll see them run and tumble, right, and do these things. So notice, did I say key Y, key W, key A? No, I didn't, right? Because all we're thinking is some chemical or light flicks a domino, and that causes the flagellum to change direction. That's all we need to understand about this, right? You may understand more about that because you understand signal transduction pathways. Well, kind of rose on your nose, that's wonderful, but you don't need it here. What we want to know is, right, that they're responding to chemicals, they're responding to light. It's not random, it's them choosing a good place to be, right? And they're choosing a good place to be based on the abundance of good stuff and the lack of bad stuff. And that can even be crowding. We're going to talk in chapter 8 about some other crazy stuff that bacteria do. Right? Like, spoiler alert, they will lie low and pretend like they're nice inside your body until they have a bunch of friends, and then they will jump you. They can actually figure that out because they're sensing things about the environment. So the idea that these animalcules are just these little things that float in the air, it's just all happenstance, they're so simple, of course they'd spontaneously generate, is really not true at all. They are as sophisticated as you are, right, in terms of being able to respond to environmental changes. Now, they might be stuck in a flask, and so you're like, well, I guess you really can't, because I could just leave you in the flask and you would die, right? But you're kind of stuck on the earth, right? It's just a bigger box. You're doing your best to respond to environmental changes in your environment, and they're doing the same thing in theirs. So they are as sophisticated as we are in terms of registering change in the environment and trying to accommodate themselves to that change, moving toward things they like, moving away toward things they don't like, right? So this is a study that was actually done on enteric prairie-trichus taxus, just in case you're like at a cocktail party someday, and you're talking about taxus, and someone's like, excuse, foolish one, no. And you're like, I'm sorry, let me just be more precise. I'm referring to enteric prairie-trichus taxus. And you're like, right? Enteric, remember we learned that word means guts. Prairie-trichus means flagella all over. So this is a study that was done on gut bacteria who have prairie-trichus flagella, like E. coli. I'm not going to ask you that, but I'm just giving you that disclaimer because not every bacterium has the same situation. But what they all do, if they have flagella, is move toward things they like and away from things they don't like. So they're not leaving life up to chance. Even in that broth, if somebody got in that broth, they would swim away from the other bacteria and try to get the good stuff all by themselves, right? They would move if they could. So that's it for that today. Don't jump up yet. I'm going to show you a picture and take roll, and then we're going to go on break. I want to show you a picture because you are going to be doing a lab today with a variety of different kinds of media. And it's easier for us to see them as pictures first, right? You're welcome to look at them on the back counter before you leave. You don't have to. You're going to get some of these and take them to your desks. But we use media primarily in this class, which are called complex media. Complex is kind of a funny word. It really means we don't really know what's inside. It's basically like chicken broth. You're like, I don't know, right? Like, you ever make a chicken broth? It's kind of a dying art, right? Unless it's Thanksgiving and you've got the turkey carcass, right? And then what do? You boil the turkey carcass, right? You put a bunch of veggies, and you take the whole carcass, and you throw it in there, and you simmer it for like 3 days, and then you throw away all the stuff, and you get the broth, right? What's in it? Stuff from turkey? I don't know. That's like complex media. But like that turkey broth, super easy to make, right? You just throw some stuff in a pot, simmer it for a while, and it's delicious, and it's good for you, and it has vitamins, right? It's wonderful. Most of what we use is complex media. Complex media is great for growing a variety of organisms. E. coli, Staph aureus, any of these things that we grow, they all grow on complex media. Today, the complex media that you're going to be using is something called tryptic soy broth and tryptic soy agar. We could have also used Luria bertani broth and Luria bertani agar, which is LB agar. We could have used nutrient agar and nutrient broth. Doesn't matter, it's all chicken broth. Turkey broth, chicken broth, beef broth. All of those things can be used to make a soup, right? Some of those things we add a inert substance called agar to. Agar actually comes from the same algae that can be used to make the wraps in sushi. Not the wraps, part of sushi, right? But it's an indigestible carbohydrate, and it's sort of like gelatin, right? If you ever made Jell-O, you make Jell-O starts as a powder. You put it in boiling water, the powder melts, and what happens when the Jell-O cools? It gets solid, right? So agar we can add to liquid media, and we get it really hot so the agar dissolves, and when it cools, it solidifies into solid media. So when we have tryptic soy broth, it just means that broth with no agar. When we have tryptic soy agar, it means it's solid, right? So solid allows us to grow bacteria on the surface of something. And that surface could be a petri dish, right? So you're going to be working with petri dishes today. Each of these is a colony, right? Where one single E. coli cell landed all by himself and grew and grew and grew and made lots and lots of babies. And now there's so many little baby E. coli that you can see them with your naked eye. We can also, right, that's a very common kind of solid media. We also have slants. And you're going to both be taking from a slant today and inoculating onto a slant today, because slants are really useful for us to just have as like maintenance cultures. If you want bacteria that you're going to be using for multiple days, we put them on slants, because slants have a big area to surface, right? So there's tons of good stuff in here that can kind of seep up into the media and feed them. So when we grow on slants, I used a pigmented organism here so you can see it. Right, you actually take your inoculating loop and you smear it all over the surface of the slant, right? Over and over and over and over and over and over and over again, all over the surface so that you can grow them, right? So this is sort of a front view, that's a side view. This is something called an agar deep. That's one we're not going to be working with today, but it is one we will be working with in our motility lab. So basically, if I take it while it's still hot and I pour it into a flat dish, that makes me a petri dish. If I pour it into a test tube and then while it's still hot, I tilt that rack on its side, they will all dry or cool as slants. If I pour it into a tube and just leave it sitting up like this, it will become a deep. And if I don't put any agar in it and I just pour that liquid into a tube, then we have a broth. So you today are going to be working with broths, slants, and petri dishes, and then next week, I believe next Thursday, you'll be working with a DEET right there. So I just wanted to show those to you because it's kind of hard to conceptualize, right? And I'm going to demo how to do those for you today, but obviously, right, it's like hard for you guys to see the difference between those two. If I hold them up to you, but if I show you a picture, it's a little easier. So that is the stuff we're going to be using today. We're going to take roll and then see what time it is and go from there. Okay, Shadi and Amy and Louis.